shouldn't take us the full 2 days, but maybe we can have a little review or answer some questions or whatever on the 19th. Got a catch-up day on the 19th. Then our test, our last exam, the final, is the 21st. Chapter 17, 18, and 20. That's our 3 chapters, and then we'll do our presentations in 2 days, and we'll take our final quiz on the 3rd. That's the plan. So, chapter 20 is our antimicrobial drugs. We talked about in chapter 6 how to grow bacteria, the stuff that's required to grow, and then in chapter 7 we talked about how to kill them, but we did not talk antibiotics. So, I don't know why they end but that's at the end. They should put it in like chapter 8, but whatever. So, antimicrobial drugs, these are, notice it doesn't say antibiotics. Antibiotics only work on bacteria. So we are going to talk about some of the drugs that are used for viruses and some that are used for protozoans. So that's why they call it antimicrobial. Chapter starts out talking about antibiotics, though. Chapter starts out talking about antibiotics. Now, as I look at my notes, I see my notes say chapter 20 antibiotics. That's a lie, I guess. So I screwed that up right at the beginning. So most of it's about antibiotics, and then slash, and some other stuff. So we've got 2 terms to define here. I think we defined these in lab. Bacteriostatic versus bacteriocidal. What's the difference?
One stops growth and one kills.
Which is which?
Static is stops.
Static just stops it from growing. And cidal literally kills. Right? It kills the organism. Bacteriostatic doesn't kill it, it just stops it from growing. So the organisms that are there are still going to be there. Hopefully they'll be removed by your immune system, but bactericidal means that the drug is actually going to attack the organism and kill it. So when we develop antibiotics, like I do it, I don't do it, but when they develop antibiotics, the goal is to target something different than human cells, right? You can't take a drug that's going to damage DNA, because then our cells will be damaged and we'll be in trouble. So we have to target something that's unique to the organism, whether we're talking about bacteria, protozoans, worms, or viruses. It's got to be different. You can't use a drug that's going to attack yourself. So the main target for most antibiotics, the target for destruction is the cell wall, because we don't have a cell wall. Does anybody remember what the cell wall is made of in bacteria? Maybe we should have a comprehensive exam.
It's not peptidoglycan?
Yes, it is peptidoglycan.
Oh.
Peptidoglycan, right? The protein sugar coat. So, bacteria have that. Whether it's a thick organism, a thick wall or a thin wall, they still have that. We don't have that, so that's a great target. If you destroy or damage the cell wall, bacterial cell membranes are not nearly as strong as humans. They don't have as much of the cholesterol type fat in there, and they're weak, and they fall apart, and the cell dies. So, lose the cell wall, organism dies by lysis. Second mechanism of action that's second most common is inhibiting protein synthesis. And there's several different drugs we're going to talk about that work at this level. So here we're going to target something that has to do with protein synthesis. And remember, ribosomes, bacterial ribosomes, are a little bit different than humans. Do you remember how they're different?
Bacterial ribosomes? They're just RNA?
Oh, the 70.
70, yes.
And then
versus the ADS, right? So they're a little bit different. They still do the same job, but we can target them because they're actually different than humans. The closer that we get to targeting something that humans have, the worse the side effects. The closer it is, the greater the chance that the drug will affect some of our structures as well. So, protein synthesis is the second most common. And as you can see here, targeting the 50S subunit or 30S subunit, remember we have a 60 and a 40, so it's just a little bit different structure. That's why we can use different chemicals to bind them up. Third most common is, well, maybe it's not, let's see. Maybe I've got it as listed as 3 for damaged cell membrane. I don't think that's the third most common, but we'll talk about it. Damage the cell membrane. So in bacteria have different types of cholesterol structure in the cell membrane than humans have. We have cholesterol. It's a fatty ring structure. Bacteria have something called sterol. So if you can mess with the sterol, which gives the cell membrane strength, you can damage the cell membrane, resulting in the death of the cell. We just got to target the different fat. We have cholesterol, bacteria have sterol. Another fourth mechanism is inhibiting nucleic acid synthesis. So here we're going to inhibit or interfere in some portion of transcription and then translation. In this case, it's very similar to us. So mostly anything that's going to work here is going to have some side effects in humans because it's getting closer and closer and closer to our structures. And then the fifth mechanism of action, which would be great if bacteria metabolize things that much differently than we do, but they really don't. Their metabolic pathways are very much like ours. But one is something called folic acid, and bacteria require folic acid, and what they do is they utilize an intermediary molecule called PABA. So PABA is used in making folic acid. We get folic acid from our diet. Bacteria produce it. They have a pathway to make it. We don't make it ourselves. We get it in our diet. So if we can block the PABA, then we can block folic acid. If the cell doesn't have folic acid, it dies because it has to have folic acid for survival. So there's a metabolic pathway that bacteria have that we don't. That's a great target because it doesn't affect us at all, right? It's a good one to target. But there aren't that many of them. There aren't that many that bacteria are unique for that don't match us. So what this chapter does is goes through several of the antibiotics that you've probably been on, you've probably heard of. It's not every antibiotic from the book. I picked ones that I'd heard of and I'd seen and maybe been on or heard of, whatever. So the most common drugs, or the common drugs that I chose to talk about, number one is penicillin. We've all heard of penicillin. Penicillin was the first antibiotic discovered. It was discovered in 1928. We talked about this in class yesterday, where a guy was growing some bacteria on a petri dish. It got contaminated with penicillium mold, green mold, and that killed the organism. It stopped it from growing. And so they figured out what molecules were involved, and penicillin became the first antibiotic. Now, because it was an antibiotic, and it was killing bacteria, it tended to be overused when it was first developed, which then allowed for the organisms to become resistant to it, which is why all organisms become resistant to antibiotics. It's the overuse. So penicillin is actually it's a group of drugs of 50 different related antibiotics. There's 50 different types of penicillin. They all have this common core. They all have this common core, this common nucleus. And the other thing that's important in this common nucleus is this little chemical structure called the beta-lactam ring. What this does is it interferes in the production of the peptidoglycan cell wall. So this gets in the middle of those lattice. And if you can't make a good strong cell wall, you can't make a good strong bacterium and the cell will blow open. So, mechanism of action for penicillin is inhibit cell wall synthesis. All penicillins inhibit the cell wall. There are, as I said, there's 50 different ones. Prevents the cross-linking of our peptidoglycans, prevents the lattice structure, weak cell wall, weak cell membrane, cell breaks open. The natural penicillins, the first ones that were discovered, were literally extracted from the mold Penicillium. They have a relatively narrow spectrum. Oh, spectral range, I didn't talk about that. Spectral range is the range of effectiveness. You've heard of antibiotics being called broad spectrum. That's a broad spectrum antibiotic. What does that mean? It's going to kill lots of different types of organs. It can kill gram-positives, gram-negatives, right? It's broad spectrum. If you go to the urgent care and you have a UTI, they will put you on a broad spectrum antibiotic because they don't know what it is. They don't know if it's gram-positive, they don't know if it's gram-negative. They'll culture it, they'll send it off to the lab to grow it and see if we can figure out what it is. They may change your antibiotic once they figure it out. But they'll start you on a broad spectrum. Narrow spectrum, obviously, is the opposite. Now we're going to target specific. We're either going to target, we may target gram-positives, we may target gram-negatives, we may target specific organisms like Pseudomonas directly, whatever. So the first natural penicillin was from the mold. And what's interesting about bacteria is some of them produce an enzyme called penicillinase. And what penicillinase does, especially staphylococci are notorious for having this enzyme, they break open that beta-lactam ring. So if they break open that beta-lactam ring, guess what? It messes up the way the drug works. You have to have that beta-lactam ring in there to get in the middle of the cross-links, to mess up the cell wall. If you open it up, it doesn't work anymore. So, staphs have this enzyme called penicillinase, which means that penicillin ain't gonna work on those staphs. Another penicillin family antibiotic, it ends in -cillin, it's in the penicillin family. It's going to have this core, is methicillin. And methicillin is a semi-synthetic penicillin, and so it's partially is the natural core, and then part of it is man-made. So what they usually do is they use the common nucleus, and then they change this out on this side to somehow try to block the way penicillinases can get in here and open up that core. Methicillin is the M in MRSA. So MRSA stands for methicillin-resistant Staph aureus. Methicillin-resistant Staph aureus. That's what it technically stands for. Really, Staph aureus should be considered multi-resistant because it's resistant to many drugs. Methicillin isn't used in the United States anymore. I understand it's still used in Europe, but it is not used, or in other parts of the world, but it's not used in the United States anymore because of MRSA. So it was used originally, it's one of the early semi-synthetic, man-made type penicillins to evade those penicillinases, and it was overused, and so there we go. Ampicillin and amoxicillin. Ampicillin is, I don't know, it's used occasionally. Often used in labs and stuff, but amoxicillin, I know you've probably heard of amoxicillin. You've probably been on amoxicillin. I don't know if I've got amoxicillin. No. Here's what ampicillin looks like. Amoxicillin is going to be similar to that structure. Amoxicillin is broad spectrum. Amoxicillin actually works on gram-positives and gram-negatives. This is a go-to for a UTI or some kind of infection that we don't know what it is. Used a lot. One of the first ones used. You've probably also heard of Augmentin. You've probably been on it. Augmentin is amoxicillin, so it's going to be considered broad spectrum. It's amoxicillin and it has a penicillinase inhibitor. So the antibiotic in Augmentin is amoxicillin. And what they've added to it, to the drug, is a penicillinase inhibitor. So it blocks the penicillinase of especially the gram-positive, and that helps it work better. Last one in this group is called aztreonam. I don't know if you've heard of this one. It is used to evade penicillinases. It is pretty specific though as far as gram-negative. It's got a narrow spectral range. They use it mostly for Pseudomonas and E. coli infections. Relatively new one, but it's gram-negative mostly. So typically, you think of penicillin family stuff as gram-positive, but there's also some some broad-spectrum drugs in there, and then this aztreonam, which is gram-negative.
Why don't those two have the penicillin ending that the rest of them?
Ask the pharmaceutical company.
But they're still the penicillin.
They're still the penicillin family.
Okay.
Yep, ask the pharmaceutical company.
Can't keep it easy.
No. Second group here is cephalosporins. They are also cell wall synthesis, so the mechanism of action is targeting the cell wall. Cephalosporins come from the mold, Cephalosporium. Many of our antibiotics come from different molds, but a whole bunch of them also come from other organisms, like actinomyces and streptomyces are a bacteria, but they produce chemicals that kill other bacteria. So a lot of those drugs come from them. Cephalosporins are similar. Look, they've got that similar beta-lactam ring, but they've got this weird circle thing that is not like the penicillin core. So the penicillin core looks like this. Here's the cephalosporin. So the structure of it helps to block the penicillinases getting in there and breaking open that beta-lactam ring. So they're more resistant to the penicillinases. Cephalosporins tend to be more effective against gram-negatives, although they are considered broad spectrum. But whereas penicillin tends to be leaning toward the gram-positives, cephalosporin is more equal, gram-positive and gram-negative. And I don't know if you've ever heard of the generations of cephalosporins. So there's multiple generations of cephalosporins. This R side is the semi-synthetic part, the part they add in the lab. And they change it because bacteria become resistant to the antibiotic. And so when they change it, then we call it the second generation of cephalosporin. They change it again, they call it the third generation. I think right now we're in the fourth or fifth generation of the cephalosporins, and they're talking about going back to the first one, because we haven't seen it for so long that hopefully the bacteria that are out there would be resistant to them. So, a little bit different core there, helping to block the penicillinases from getting in there and breaking open that beta-lactam ring. Next one is polypeptide antibiotics. These are small proteins. Again, mechanism of action is inhibiting cell wall synthesis. It is again cell wall synthesis. So, two of the antibiotics that I know you've heard of that are polypeptide, they're short proteins. One is called bacitracin.
Favorite cuts.
Bacitracin and vancomycin. Vancomycin is kind of the big dog of antibiotics. So bacitracin is actually a polypeptide antibiotic that comes from the species, the genus Bacillus. It actually comes literally from Bacillus subtilis. So Bacillus subtilis makes bacitracin. It makes the polypeptide. And again, screws up the linear strands of our lattice cell wall, inhibiting the cell wall synthesis. Bacitracin got its name because it was discovered in a wound of a girl. So a girl had a wound. I believe it was a skin wound. And there was something in the wound that was, I don't know how they figured this out, something in the wound that was killing other bacteria. And they purified it, realized that it was coming, somehow they figured it out, realized it was coming from a bacillus. And so bacitracin got its name because the girl's name was Tracy. And I'm not bullshitting you, this is true. So bacit for bacillus, tracin to honor the girl. So it came from a wound, from a bacillus in a wound from a girl named Tracy. And that's the truth. Honest fact. I can't remember if Tracy is her first name or last. I think it's her last name. I can't remember. But anyway, that's Tracy. Another one that is a polypeptide antibiotic coming from a small, short protein is called vancomycin. And vancomycin is usually used as the last line of defense for MRSA or resistant Staph aureus infections. It is gram-positive only. It is gram-positive only, as is bacitracin. Bacitracin is gram-positive only, works best on staphs and streps. Vancomycin is gram-positive only. It's too big to get into the gram-negative cell wall, so it doesn't work at all on gram-negatives. Gram-negatives are resistant to vancomycin. But vanco should be the last resort for staph infections because it's the last thing that is working well on staph infections, resistant staph infections. So what you want to do is somebody's got a staph infection, or you think it's a staph infection, start with some other antibiotics first, see if it's going to work. If it doesn't work, then move to vanco. Because if you start with vanco, and everybody starts with vanco, then guess what? We're going to get resistant to vancomycin. And at the end of the chapter, we'll talk about how bacteria develop resistance. It's not like they just say, well, I don't like you antibiotic anymore. I'm going to become resistant to you. There are mechanisms that go along with it. And then if you don't have vancomycin, then we're really running out of antibiotics to treat MRSA type infections. I know.
I know you always hear it.
It's used for C. diff.
Use a lot.
I know.
Yeah.
I know.
Oh.
Well, so we're gonna use it for C. diff. Guess what we have now running around?
Yeah.
Vancomycin resistant enterococcus, which is a gut organism. It's a gram-positive, we've worked with enterococcus, right? It's gram-positive coccus from the gut. You pile people with antibiotics, you're gonna get resistant. And now, with VRE, we don't have a ton of drugs to treat it. And then what do you do? That's the problem we're running into. Antimicrobial agents, this is also cell wall, but it's a different type of cell wall. This is for acid-fast organisms. Remember, acid-fast organisms have that mycolic acid, that waxy substance. So these are for treating mycobacterial infections like Mycobacterium leprosy, Mycobacterium tuberculosis, those kinds of infections. And what it does is it messes with the waxy wall which ultimately damages the cell. Interesting thing about mycobacterial agents, those drugs, is that you have to take these for months because the generation time for mycobacterium is up to 10 days. So, you know, a typical course of antibiotics is what, 3 days, maybe a week, right? Well, if it takes 10 days for an organism to divide once, how much damage are you gonna do in a week? Not much, right? So if your antibiotic regimen is done in a week and the bacteria are still there dividing, because it takes 10 days to divide, then it's a problem. So you have to be on these for months, sometimes years, because of such a slow growth time. Okay? Number 5, 1 through 5 are all cell wall. 1 through 5 are cell wall mechanism, cell wall synthesis. I lied, 1 through 4. Sorry, 1 through 4. 1 through 4 are cell wall synthesis. Pardon me. Number 5 is chloramphenicol. This is our first protein synthesis target. Mechanism of action is disrupting protein synthesis. The closer it gets to us, the more side effects and toxic it is to us. Chloramphenicol is broad spectrum, but it tends to be highly toxic. Highly toxic. It's used in certain diseases because it's very small and can diffuse easily. So it's commonly used for intracellular organisms, organisms that grow inside cells, like rickettsia. Rickettsia causes Rocky Mountain spotted fever. That's an organism that lives inside your cells. So if it's inside your cell, how are we gonna get to it?
Inside.
Well, chloramphenicol is small enough to enter the cell membrane of our cell. Chloramphenicol can also cross the blood-brain barrier, so you can treat brain abscesses, but it tends to be quite toxic. And its main toxicity is that it suppresses bone marrow activity. And what happens in your bone marrow? You make your
White blood cells.
and platelets. You make red, white cells and platelets. So that would suck. Damage your immune system, become anemic, and not be able to clot. So it'd be tough. But we use it in certain cases because you got a disease you got to try to fight. Five through 8 are protein synthesis. 5 through 8 are protein synthesis. 1 through 4, cell wall. 5 through 8, protein synthesis. Next one's called neomycin and gentamicin. Neomycin is topical. Anybody have any neomycin at their house? Mm-hmm. What is it? It's a salve. Put it on owies, right? Inhibits protein synthesis. Obviously the topical isn't going to cause that much damage to you because it's topical, but like any oral ones like gentamicin can cause damage to the auditory nerve. It can affect your hearing. These are both broad spectrum. Both broad spectrum. Gentamicin tends to be used for Pseudomonas infections in cystic fibrosis patients. But it is considered broad spectrum. And it binds to the 30S ribosome, 30S subunit ribosome, so it can't translate your proteins. But it can affect your hearing. Then there's tetracyclines. Maybe you've heard of tetracyclines, again, inhibiting protein synthesis. This one actually blocks the tRNAs from binding. Another broad spectrum, both gram positive and gram negative. Tetracyclines are used for a lot of STDs. They're also used for acne. Some people go on tetracycline for acne. If you are a female and and you are on tetracycline for acne, every month you have to have a pregnancy test, because tetracycline crosses the placenta and causes damage to the fetus. It specifically makes the teeth turn gray. So you don't want that to happen. I had a student once, we were talking about this, and she says, yeah, I'm on tetracycline and I have to be on, even though I'm on birth control, I have to go get pregnancy tested every month.
Does
So
he like
that's-
on 2 forms of birth control or something?
What's that?
Don't you have to be like on 2?
I don't know. I don't know about that. I don't know.
I think you just have to take one layer for the test. And then they tell you to use protection.
There you go. Because some acne is caused by bacteria in the follicle. Overgrowth of bacteria. Last one is erythromycin. You may have heard of this one. This one is an alternative to penicillin. So if you're allergic to penicillin, they'll probably put you on erythromycin. Erythromycin inhibits protein synthesis, but it's only gram-positives. Targets gram-positives only. Go-to for people that are allergic to penicillin. And I put erythromycin on here because I thought it was pretty. Pretty chemical structure. Kind of reminds me of like a big fancy snowflake, or you could hang it on a necklace. I just like it. You don't have to know your chemical structures on these. Number 10 and 11 are nucleic acid synthesis. Sorry, gotta do number 9. Number 9! I'm doing really good here today so far. Number 9 is cell membrane. It's the only one I have in here for cell membrane. It's called polymyxin B. Polymyxin B damages the cell membrane. It's a topical ointment, and it is good for gram negatives. So, you maybe have polymyxin B in your cupboard and you don't even know it. Have you ever heard of, or do you ever buy, or do you have it in your cupboard, triple antibiotic? That's what I use. Triple antibiotic. It was my dad's favorite. He must have had 10 bottles or 10 tubes of triple antibiotic. Triple antibiotic has bacitracin, neomycin, and polymyxin B. Polymyxin B, mechanism of action is cell membrane, spectrum, gram negative. What was neomycin? Neomycin was protein synthesis and broad. And what was bacitracin? Cell wall. Cell wall and target. Oh my gosh, look at that. We got the whole spectrum covered. Triple antibiotic is the better antibiotic choice for skin wounds because you got everything covered. Sometimes, like if you get bacitracin, it is bacitracin only. Neomycin as a brand name usually has all 3. Triple antibiotic is like the Walmart brand, you know, the generic thing, but it's got all 3.
If I just use plain bacitracin on my girls, they get blisters, but if I use triple antibiotic, they don't. I don't know why.
I don't know why either. But this is a better choice anyway. Just throw your bacitracin in the garbage. You don't need it. Yep, best choice. Polymyxin B. Probably never heard of it, but it's probably in your cupboard. Okay, that's our only cell membrane one. 10 and 11, nucleic acid synthesis. messing with nucleic acid. Number 10 is rifampin, and it inhibits the mRNA synthesis, blocks mRNA. It binds actually to the RNA polymerase. Very small, can cross the blood-brain barrier, can enter tissues. Typically used for tuberculosis and leprosy. It is gram-positive only though. So you can use it for waxy ones and gram-positive. It's not good for gram-negative.
Typically used for, you said,
Leprosy and TB. The last one includes quinolones as well, and this should say ciprofloxacin. I have this spelled wrong. Ciprofloxacin, but mostly it goes by Cipro. You've probably heard of Cipro. Put Johnson, Cipro. Oh. Ciprofloxacin, or Cipro. These inhibit DNA gyrase. DNA gyrase unwinds the DNA for replication, so it blocks that enzyme from doing that. Broad spectrum. Used to treat anthrax, but used to treat lots of stuff. Used to treat UTIs. It's a common one for UTI go-to. But if you stay on it for a long period of time, it can cause damage to your tendons and result in tendinitis and rupture of your tendons. You don't want to stay on it for too long. You want to go back to it all the time. When my mom had her big kidney stone, remember the story from anatomy? Her big kidney stone, and the antibiotics, wouldn't reach the middle of it, so she kept getting recurrent UTIs because this stone was all porous and in there and stuck. And we were going to get her kidney removed at Mayo, but I had to be around my schedule, and the doctor's schedule. So the first time we could get it done was in December, and we discovered it in July. So they put her on antibiotics off and on and off and on and off and on until finally she went on Cipro continuously every day for 3 months till we got her to Mayo Clinic. Probably not a good idea, but she wasn't running any marathons or anything, so she didn't rupture any tendons. Last one. This is our metabolic pathway, disrupting the metabolic pathway. Or inhibiting metabolites, messing with the metabolic pathway. And these are sulfonamides. They are bacteriostatic. So they're not bactericidal, but they stop the growth. And as I said, they compete with them, they bind to or block an intermediary component in this pathway called PABA, and that makes the organism unable to make folic acid. So without folic acid, the cell can't survive because it's required for their survival. The one that you've probably heard of, or maybe not, is called trimethoprim sulfamethoxazole. They work hand in hand, they work synergistically, they both do the same thing, but together they work better. This is another go-to. This is another one it's broad spectrum, so it's a kind of a go to this one. Its brand name is called Bactrim. Have you ever heard of Bactrim? That is this guy. Generic would be our trimethoprim sulfamethoxazole. Let's see, somebody asked me once about a Z-Pack. Oh, we're gonna put me on a Z-Pack. Azithromycin. It's actually inhibits protein synthesis. It's a protein synthesis one. And I'm guessing it's broad spectrum, I don't have that on here, but I'm guessing it's broad spectrum because it's a go-to for whatever. I've never been on a Z-Pack. Huh? No, I've never been on a
7-day course, right?
It's, I think it's only like 5 days, like, the first day you take 2, and then it's,
Okay.
you get like 3 or 4 days of just 1 pill.
Mm.
I remember my mom had a sinus infection, and they put her on like a long action, she's like, they don't know what they're doing, just give me a Z-Pack, and like, Z-Pack won't work.
I haven't been on knock on wood, I haven't been on an antibiotic in, God, years, probably 15 years or more.
The army hands it out like candy.
They shouldn't.
Unfortunately, so do urgent cares and stuff. All right, so that's your grouping for antibiotics. You've got 5 mechanisms of action, and you've got some different spectral ranges. Antifungal drugs. Remember, fungi are eukaryotes, so they're more like us. So the more they are like us, the harder it is to treat them. There are fewer antifungal drugs than there are antibiotics, just because there's lesser targets. Some of them you've probably heard of. Some of the antifungal drugs damage the cell membrane. Again, in fungi cell membrane, they've got a cholesterol type protein, a fat. We have cholesterol, bacteria have sterol. They have something called ergosterol. So when you mess with that, you mess with the cell membrane, you make it weak, it falls apart. Amphotericin B is one. I don't know if you've ever heard of it. Amphotericin B. It treats fungal infections, but it's toxic to your kidney. So, again, the more like us, the organism, the fewer the targets and the more the side effects. Second one in this group is chlorotrimazole or miconazole. This is like the sprays that you put on your feet for athlete's foot, or the ointment stuff for yeast infections, vaginal yeast infections. Target is the cell membrane. And then some drugs target fungal cell walls. Fungi can have a cell wall too. It's made up of a sugar coat called glucan. And if we can mess with the cell wall, we can mess with its ability to stay together and cause lysis. I don't have a drug for that one, but that's the other target. That's about it for the targets for fungi.
So is miconazole the same thing as like fluconazole? Does that fall into the same?
It falls in that ZOLE, so it's gotta be. Whenever the antibiotics end in the same thing, or the drug ends in the same, like, it's of the same class. I'm guessing yes. What's it used for? It's gotta be
For yeast
Yeah.
infection.
I mean, there's different ones for feet and.
I think the fluconazole is just an oral.
Yeah,
Is
just
it
the one pill.
Yeah, so that's oral and then it's for creams.
Then there's antiviral drugs, and there aren't many of these because viruses like to mutate, and then things don't work on them like they should. We've got some antiviral drugs like acyclovir. Acyclovir uses it's a, you know the G's, A's, T's, and C's in your DNA? It's a different G. And the virus incorporates, or in the assembly and the transcription translation of the virus, for whatever reason, the weird G gets incorporated, but our cells don't. And so it helps to stop its replication. Acyclovir is for herpes infections, mostly. It's the weird G that doesn't bind to the DNA or the sugar, so it can't continue its replication process. Some of the anti-HIV drugs, the AIDS types drugs, like antiretroviral drugs, they block reverse transcriptase. There's lots of different ones for HIV, they block binding to the receptor, they block reverse transcriptase, they're a protease inhibitor, so that when the proteins for the virus are made, they end up being made a little bit too long and they get clipped by protease. And if protease can't clip, then the virus can't be assembled like it's supposed to. So it blocks continued production. And then neuraminidase, that is what is used in our influenza, our H and our N. Remember we talked about that? H5N1, H1N1, H3N2, whatever. H was for the binding, N was for the release. So this is like Tamiflu. The neuraminidase inhibitors are like Tamiflu. If you get influenza or you're exposed to somebody with influenza, you can take Tamiflu or Relenza, that's the other one, right? Within the first 48 hours, and it helps block the release of the virus from the cell, which then decreases the amount of virus, which decreases your symptoms. It doesn't cure it, but it helps reduce it. None of these are curing. They really aren't curing. Even acyclovir doesn't cure herpes. Herpes lives in your neurons, and the only time that we can, well, I guess that works inside cells, but it doesn't kill the virus, because remember, herpes is incorporated into your DNA, so you can't get rid of it in your DNA. So that's about it for our antiviral drugs. And then we've got our antiprotozoan and anti-helminth drugs. Some of these have very serious side effects because they're even more like us. Like worms, they're multicellular. They're even more like us. The other side effect that can happen, especially with like a worm infection, is if you kill the worm, and you got all these worm pieces and parts floating around, you can develop allergic reactions, deadly allergic reactions to these worm parts, these dead worm parts. So that's not good. Just don't get a worm infection and then you'll be okay. Mechanism of action of these is blocking anaerobic respiration. In other words, blocking the glucose uptake. A lot of these live in your gut. And in your gut, there's not a lot of oxygen, so they undergo anaerobic respiration. So if you can block their ability to make ATP, they can't have energy to survive. Some drugs in this group block the cytoskeleton, which then inhibits the absorption of their nutrients. So if they live in your gut, they like to steal your nutrients. So if we mess with the cytoskeleton, the microtubules of the worm or protozoan, they don't get to eat and they'll starve to death. And then my favorite mechanism of action of all time for whatever is unknown. Mechanism of action is unknown. I don't know how it's going to work, but it seems to do good, so just take it and it'll be fine. I hate that. I hate it when the mechanism of action is unknown. If you don't know the mechanism of action, don't put it in somebody's mouth. That's my opinion.
Then how did that pass clinical trials?
They've got a suspected mechanism of action.
So they're just like, we think this
And
is
as long as it doesn't cause massive problems to the person, then it's okay.
Nothing.
Yep. So, worm drug. Have you ever heard of it? It's called ivermectin. It's used for treating worm infections in cattle and like lice on their skin, on their hair. And so if a cow has lice, which I guess is common, you spray them down with ivermectin in a spray form, or you give them liquid ivermectin if they've got worms. It's a deworming.
And for COVID.
What?
It's the one that we were talking about in
Yes, Sierra, yes. Yes.
Yeah.
So, here's the scoop with ivermectin. It's used for worms, scabies, lice, in livestock. And what it does is it basically paralyzes the worm. It actually comes from a bacterium. It's an antiviral that came from a Streptomyces bacterium. And not that this matters, but it was discovered near a golf course in Japan. Whatever that matters, I mean, that's where they discovered it. And the reason that ivermectin got so blown out of proportion with COVID is that there was some evidence, there was some research that had been done that suggested that ivermectin was an antiviral. And in the Petri dish, it had antiviral capabilities and actions. But it could never be transferred to cell culture or mice, living organisms, and so the research ended there. It didn't work in a mouse, it didn't work in a living organism. It showed like maybe it worked in a Petri dish at very, very high concentrations. So the ivermectin people got a hold of this article, and you heard the stories. Their family member got COVID, and they insisted that the medical facility gave them ivermectin, and the facilities are like, no, that is not proven. It is, in fact, it is basically disproven in living organisms. There's no guidance for dosage.
We've actually had a patient like drink the cat.
Oh, yeah. Yes. I had a student last semester whose, was it her grandpa or uncle? He's a rancher. She grew up on a ranch by faith. And he's a rancher, and every morning he would go out to his liquid ivermectin and take a shot. And then he started having some, things were weird. Like he was having some cognitive issues, and I don't know whatever ended up happening, but she kept telling him to stop doing this, and he wouldn't do it. He did it prophylactically. If he just drank his ivermectin every day, like his orange juice, then he would not get infected with COVID.
I just couldn't imagine drinking that stuff.
Yeah, it's a talk.
Safe doses for worm infections in cows is 20 to 80 nanograms. Nanograms. In the Petri dish, the antiviral capability, the level of amount of ivermectin that was needed to work on viruses was in the milligram range. It was 1,000 times more than what they use to kill worms in cows. And so you'd have to use 1,000 times, you'd have to drink the whole gallon of stuff to have this same effect. And that's just not, no. No, that is not safe. Not safe at all. Anyway, that's the story of ivermectin. You read the story, and I think this was somebody in, I think it was in South Dakota. Maybe it was Minnesota or Iowa or somewhere like that, but it was somewhere in the neighborhood. And her husband got COVID, and he was in the hospital and vented. I mean, he'd been there for months and sick. And she insisted, although it didn't say in the article what medication she wanted, she insisted that the facility gave him unapproved COVID medications. And the hospital said, no, we can't do this. She found somebody in Texas that agreed to do it, to give the whatever it was, I'm guessing it was ivermectin. She got him life flighted down to Texas. She got the medication she wanted. Guess what unfortunately happened to the husband anyway? He died anyway. And then she blamed the fact that it was too late.
There's a lady at Monument who I think was mad because they weren't giving her husband
Oh,
that,
I know.
and she boycotted. Like she's out there with signs boycotting Monument because of it.
Okay, where are you gonna go? I mean,
Right.
it's the only show in town. There's no other hospital in the region that is not Monument. So if you're gonna get sick, you better be in Denver or something else or something, because you got no other choice. All right, last thing, and then this is what we did in class yesterday. This is testing our antibiotics, and this is our diffusion method. So we do this, labs do this to test for effectiveness of an antibiotic on an organism. So just like we did yesterday, we plated our lawn, you know, nice and tight. What would happen if you plated your lawn like this?
You have a bunch of gaps.
So, it'd be pretty,
You would get nice circles like that.
and then what?
You can't measure your
You
distance.
can't measure to see how well your effectiveness is, right? So we want a nice, pretty lawn. So we did that, we put our little antibiotic discs on it, and then we stuck it in the incubator. Most of yours looked good. I don't know why the Staph aureus didn't grow well, so I've got that still cooking upstairs, but I don't know why. Hopefully it'll be fine by next week. So then what we're gonna do is we're gonna come in next week and we're gonna measure the diameter of the circle, which is called the zone of inhibition, and see how that falls within the range of a chart. And depending on the size of the zone, that will tell you if your antibiotic is a good one to use for your infection. Is your organism sensitive to it, or is it resistant to it, or is it somewhere in between? And that's important to know if we need to really treat an infection aggressively, or I don't know exactly what we're working with. The other thing that we can test is the broth dilution test, and what this does is basically gives us kind of a dosage level. It's the minimum bactericidal concentration of a drug. And there are parameters, specific parameters for it, but I'm gonna kind of make something up here. So what we do is we take some small little wells or cups, and we put bacteria in it. And we want to use the same amount of bacteria. So let's say we put 1 million organisms in each of the wells. Okay, so each one has 1 million. Then what we want to do is we want to add the antibiotic to each of the wells at differing amounts and see at what level does it actually kill. So let's say here, let's say this is 1 milligram, we put 1 milligram of the drug there, and here we're gonna go 1/2 milligram, and here we're gonna go 1/4 milligram, and here we're gonna go 0.125 of a milligram, and here we're gonna go 0.63, something like that, 0, and here we're gonna go 0.03. Something like that. So we're going to decrease the drug over the course of the wells. And then we stick it in the incubator and we grow it. And let's say there's no growth here, and no growth here, and no growth here, and no growth here, but we finally have growth here and growth here. What's my minimal bactericidal concentration? Right here. That's the minimum amount it takes to kill 1 million organisms in this example. So it helps to determine dosages. At least places to start. Places to start with dosages. It's not that it's used, in people or anything like that, but it's kind of a place to start for dosages. Story about antibiotic disc diffusion method. So those of you that have been in my classes I've heard lots and lots of stories. Most of them are about my husband, because he's got great stories to tell in class. Some about my son, and I never tell about myself, but I'll tell this one about myself. So this was, geez, like I said, about 15 years ago, maybe more. Got up in the morning, went to the bathroom, thought, it's kind of burning. Maybe I'm just dehydrated. Oh well. So I went to get my coffee, looked at the newspaper, whatever I was doing, drank some coffee. About 20 minutes later, I thought, I have to pee again, and that's kind of early, fast, I have to pee again. Went back and kind of burning. I thought, oh, come on, really? So, went through the day, ignored it, drank a lot of water, just spent a bunch of time in the bathroom. So I drank a lot of water. So urgency and burning. What am I brewing? UTI. Right. So I get up the next morning, it hasn't gone away. I just thought it would miraculously go away. Hadn't gone away. So, came to school and guess what I did? Of course I did. Absolutely. I got my little yellow cup, I peed in my cup, and I put it on a Petri dish. Which one do you think I streaked it on? Close. EMB. Cause why? Cause what? Okay. What's the most common cause of UTIs? E. coli. What does E. coli look like on EMB?
The green sheet.
Oh, bright with green sheen. So I took my plate, streaked for isolation on my EMB, stuck it in the incubator, came back the next day, and pretty, bright with the green sheen. So I said, well, son of a bee, I have a UTI. So I called my doctor. Well, then I also, in Phys, we do the little pee dipstick test, which you guys are going to do here too. And it was positive for nitrites, which means bacteria. It was positive for white blood cells. It was positive for blood. I mean, the whole thing's there, you know. So I called my doctor, and I said, I have a UTI, and I know it's E. coli. Can you just give me an antibiotic? Oh no, you have to come in and pee in our cup. I said, I already know the results. I already know it. So I went in, you know, paid my money for their dipstick test, although I'd done it myself. Oh, yep, you got a UTI. Oh, no shit, I already know that. So the doctor comes in, and I told him the story, because he knows me and what I do. And I said, I know it's E. coli, I already know. And so puts me up on the table, and he said, well, I thought put you on Cipro for 3 days, and then he starts pounding on my back, and it like hurt. I went, oh. He said, does that hurt? And I said, well, yeah, kind of painful. He said, well, we're gonna give you a little longer, because guess where it had worked its way to?
Your kidneys.
My kidneys. Because I sat on it for 3 days, because I wanted to grow it. I hadn't done anything about it. So, he said, I'll put you on Cipro for 7 days. Fine, whatever. He said, can you do an antibiotic sensitivity test at school? And I said, well, yes, I can. He said, what I want you to do is test your organism with Cipro. If it's sensitive, stay on it. If it's resistant or not sensitive, call me and we'll change the antibiotic. I said, well, I can do that.
How cool.
So I came to school and I did my little lawn, put a Cipro right back in the middle of the plate, came in the next day, and big old zone, it wasn't that big, it was a big old zone, which said
Stay on it.
I was on a good choice, got rid of it. That's the last time I've been on antibiotics. Knock on wood. I don't want to see that again. So there's the end of my story. All right, last little bit here. Jeez, we got through this whole chapter. I can't believe it. What are we gonna do on?
Review.
Okay, fine.
Which one?
Otherwise, we can just take the test and start presentations early.
Thank you.
Fine, I'll come up with something to do in class on Tuesday. Alright, last section, drug resistance. How do, I told you, bacteria can become resistant to antibiotics. Does the antibiotic become resistant to the bacteria?
No, the bacteria.
Yeah, the antibiotic isn't resistant to the organism. That makes no sense, right? So, what they can do, they got 4 mechanisms. One is they literally destroy or inactivate the drug. So, like penicillinase. Penicillinase is an enzyme produced by the organism that chops the beta-lactam ring, making the penicillin completely useless. If that enzyme, if that penicillinase is on a plasmid, what does that mean? Here's the main chromosome, here's the plasmid. If the penicillinase is on the plasmid, what can happen? We can grow a sex pilus, and now he too can be penicillinase positive and resistant to penicillin. Some drugs block the penetration, some organisms block the penetration of the drug, so they block it from getting in. Some organisms can alter the target site for the drug with like antigenic variation, or they alter something in the bacteria that affects the antibiotic working on it. So there's some kind of a genetic change that changes the protein, so then the drug can't work. So it's a genetic manipulation of the organism, or a genetic mutation of the organism. And one thing that is I don't know if it is the most common, but happens, is that the organisms can literally pump the drug right out of the cell. So the antibiotic enters, and the organism has this, what's called an antibiotic efflux pump. And so the antibiotic comes in, and the bacteria just pushes it right back out. So it can't do its job. So it comes in, and it spits it right back out. All of these are genetic things. And remember, a lot of them are going to be on those plasmids. Remember, plasmids have that DNA that is not life-sustaining, but it's got extra special something, right? Like toxins or antibiotic resistance genes, like this kind of stuff. Then if you can pass it on to your neighbor, then lots of organisms can be resistant to antibiotics. And the overuse of antibiotics increases the resistance of these strains. It increases these strains. It makes them be able to grow better in the presence of the antibiotic. They develop some kind of a resistance, one of these mechanisms. And whether or not it gets sent from a plasmid from one cell to another, or it's just a genetic alteration, or they already have it because they have penicillinases, things like that. The more you expose organisms, especially your normal flora, to the antibiotic, the greater the chances the organism will be able to develop these mechanisms and the antibiotic doesn't work. So, if you have a cold, well, let's do it this way. 30% of antibiotics for an ear infection are unnecessary. 30%. Because 30% of ear infections are caused by viruses. And do antibiotics work on viruses? No. So how come you take your kid in for an ear infection, what do they give them? Antibiotic. you treat the 70%, even though a big percentage, 30% or more, is viral induced. So now we're exposing kids to antibiotics from the beginning.
Can they test for if it's,
No, it's in the middle ear.
right, that's what I was like,
50% of antibiotics are unnecessary for a sore throat. Because what are most, at least half of the sore throats? Those shooters, your NK cells out there, the virus is blowing up your cells. What's the first sign, so you know, the first symptom of a cold? Just because you have a sore throat doesn't mean it's strep. 100% of antibiotics are unnecessary for the common cold, because all colds are caused by viruses. So if you go to the urgent care, or your doctor, or whatever, with a cold, and you walk out with a prescription for an antibiotic, shame on you and shame on them. Because the more you expose your normal flora to antibiotics, the more this happens, which is why we have MRSA. It's why we have VRE. It's why we have resistant Klebsiella. It's why we've got these scary, which I'll tell you about some of these on Tuesday, antibiotic resistant bacteria that we've got some out there in the world that nothing, we don't have a drug for it. There's no drug. Oh yeah, we can go to Georgia and get the bacteriophage, right? Unless you're so sick you don't make it. I mean, so it's the reason we have these these resistant organisms is because we have overused antibiotics since the beginning of the development. Just give them an antibiotic. It's okay, it's not a big deal. Well, now we know. We've kicked ourselves in the butt by doing it.
so if you know now, why would they keep handing out antibiotics?
You know,
Like,
the attitude is changing. The attitude is changing as far as doling out antibiotics. Am I right, Brandon?
Yeah, they didn't used to, teach that
Yeah.
either. So
Right.
a lot of the older physicians will still prescribe antibiotics or,
And now they're really, really focusing on,
well, now they're really cracking down on if you have a UTI, you have to come in and get, like, a urine culture, and
right.
some doctors won't even give you antibiotics until your urine culture's back.
So
They know what,
my sister and I helped take care of this blind friend, and she got rotavirus. So she was throwing up and having diarrhea, and she's blind. And she called the ER, she called the ambulance and went to the ER. She spent almost a week in the hospital. Anyway, in the ER, they did a UA. So I'm reading the whole chart, because I'm her medical proper training, so I was reading the chart. They did a UA. It had protein in it. That's the only thing that was off. It didn't have white blood cells. It didn't have nitrites. It was not indicative of a UTI. But they put her on antibiotic regimen anyway. And there was no analysis. There was no re-UA test. There was no culture. Like, why? Why? And now she's got another one. So she's getting these chronic UTIs. Does she have a resistant organism? Possibly. She's been on freaking antibiotics every month for every other month. And they put her on an antibiotic, and I just was like, what in the heck? And that was in the ER.
could protein show up in the urine because the kidneys aren't functioning right?
I don't know. She's got type 2 diabetes.
well, a lot of people with diabetes show protein.